This video is about genetic code and other related topics about genetic code. So, encoding genetic information. Once the structure of DNA had been revealed in 1953, it became evident that the sequence of amino acids in a polypeptide was specified by the sequence of nucleotides in the DNA of a gene. It seemed very unlikely that DNA could serve as a direct physical template for the assembly of a protein. Instead, it was presumed that the information stored in the nucleotide sequence was presented in some type of genetic code. With the discovery of messenger RNA as an intermediate in the flow of information from DNA to protein, attention turned to the manner in which a sequence written in a ribonucleotide alphabet might code for a sequence in an alphabet consisting of amino acids. So, the properties of genetic code. One of the first models of genetic code was presented by the physicist George Gamow, who proposed that each amino acid in a polypeptide was encoded by three sequential nucleotides. In other words, the code words or codons for amino acids were nucleotide triplets. Gamow arrived at this conclusion by a bit of armchair logic. He reasoned that it would require at least three nucleotides for each amino acid to have its own unique codon. Consider the number of words that can be spelled using an alphabet containing four different letters corresponding to the four possible bases that can be present at a particular site in DNA or mRNA. There are four possible one letter words, 16 like 4 raised to 2 possible two letter words, and 64 like 4 raised to 3 possible three letter words. Because there are 20 different amino acids or words that have to be specified, codons must contain at least three successive nucleotides or letters. The triplet nature of the code was soon verified in a number of insightful genetic experiments conducted by Francis Crick, Sidney Brenner, and colleagues at Cambridge University. In addition to proposing that the code was triplet, Kimnow also suggested that it was overlapping. Even though the suggestion proved to be incorrect, it raises an interesting question concerning the genetic code. Consider the following question of nucleotides like AGC, AUG, GCAU, CGA. So, if the code is overlapping, then the ribosome would move along the mRNA one nucleotide at a time, recognizing a new codon with each move. At the preceding sequence, AGC would specify an amino acid, GCA would specify the next amino acid, and so on. In contrast, if the code is non overlapping, each nucleotide along the mRNA would be part of one and only one codon. In the preceding sequence, AGC, AUG, and GCA would specify successive amino acids. So, a conclusion as to whether the genetic code was overlapping or non overlapping could be inferred from studies of mutant proteins such as the mutant hemoglobin responsible for sickle cell anemia. In sickle cell anemia, as in most other cases that were studied, the mutant protein was found to contain a single amino acid substitution. If the code is overlapping, a change in one of the base pairs in the DNA could be expected to affect three consecutive codons and therefore three consecutive amino acids in the corresponding polypeptide. If, however, the code is non overlapping and each nucleotide is part of only one codon, then only one amino acid replacement would be expected. This and other data indicated that the code is non overlapping. Given a triplet code that can specify 64 different amino acids and the reality that there are only 20 amino acids to be specified, the question arises as to the function of the extra 44 triplets. If any of all of these 44 triplets code for amino acids, then at least some of the amino acids would be specified by more than one codon. A codon of this type is said to be degenerate. As it turns out, the code is highly degenerate as nearly all of the 64 possible codons specify amino acids. Those that denote 3 of the 64 have a special punctuation function. They are recognized by the ribosome as termination codons and cause reading of this message to stop. So, the degeneracy of the code was originally predicted by Francis Crick on theoretical grounds when he considered the great range in base composition among the DNA of various bacteria. It had been found, for example, that G plus C content could range from 20% to 4% of the genome, whereas the amino acid composition of the proteins from these organisms showed little overall variation. This suggested that the same amino acid were being encoded by different base sequence, which would make the code degenerate. Then identifying the codons. By 1961, the general properties of the code were known, but not one of the coding assignments of the specific triplets had been discovered. At the time, most geneticians thought it would take many years to decipher the entire code, but a breakthrough was made by Marshall Nirenberg and Heinrich Mattei, who used an enzyme polynucleotide pyrophosphoride phosphorylase to synthesize their own artificial genetic message and then determine what kind of protein they encoded. The first message they tested was a polyribonucleotide consisting exclusively of uridine. The message was called poly-U. When the poly-U was added to a test tube containing a bacterial extract with all 20 amino acids and the materials necessary for protein synthesis like ribosomes and various soluble factors, the system followed the artificial messenger's instructions and manufactured a polypeptide. The assembled polypeptide was analyzed and found to be polyphenylalanine, a polymer of amino acid phenylalanine. Nirenberg and Mattei had this shown that the codon UUU specifies phenylalanine. Over the next 4 years, the pursuit was joined by a number of laboratories, and synthesis of mRNA were constructed to test the amino acid specifications for all 64 possible codons. The result was the universal decoder chart for the genetic code, which is shown here. And the decoder chart lists the nucleotide sequence for each of the 64 possible codons in an mRNA. Instructions for reading the chart are provided. So I will be reading along with that. Ribosomes always identify an EUG as a codon to start polypeptide synthesis. In these early experiments with synthetic polynucleotides, the steps using bacterial extract to synthesize polypeptides were carried out at unusually high Mg2 plus concentrations, which allowed the ribosomes to initiate translation at any codon. So we could see first letter UCAG, along with that second letter as a row, first row UCAG, then third letter as the last column. Okay, so this is the genetic code. This universal decoder chart lists each of the 64 possible mRNA codons and the corresponding amino acids specified by that codon. To use the chart to translate the codon UGC, for example, find the first letter U in the indicated row on the left. Follow that row to the right until you reach the second letter G indicated at the top. Then find the amino acid that matches the third letter C in the row on the right. So UGC specifies the insertion of cysteine. Each amino acid except 2 has 2 or more codons that order its insertion, which makes the code degenerate. We learned what is degeneration. Degenerate code. So a given amino acid tend to be encoded by related codons. This feature reduces the likelihood that base substitutions will result in changes in the amino acid sequence of a protein. Amino acids with similar properties also tend to be clustered. Amino acids with acidic side chains are shown in red, those with basic side chains blue, those with polar uncharged side chains is green, and those with hydrophobic side chains in brown color. As discussed in the following section, decoding in the cell is carried out by tRNA, a few of which are illustrated schematically in the right side of the region. This figure, right side of this figure, tRNAs like UAA, UAG, and UGA are read as stop codons. Clustering reflects the similarity in codons that specify the same amino acid. As a result of this similarity in codon sequence, spontaneous mutations causing single base changes in a gene often will not produce a change in the amino acid sequence of the corresponding protein. So, A change in nucleotide sequence that does not affect amino acid sequence is said to be synonymous, whereas a change that causes an amino acid substitution is said to be non-synonymous. These two types of mutations are very important, which describe several of the different types of mutations. Then synonymous changes are much less likely to change an organism's phenotype than the non-synonymous changes. So consequently, non-synonymous changes are much more likely to be selected for an agonist by natural selection now that we have sequenced the genomes of related organisms, such as those of chimpanzees and humans. We can look directly at the sequences of homologous genes and see how many changes are synonymous or non-synonymous. Genes possessing an excess of non-synonymous substitutions in their coding regions are likely to have been influenced by natural selection. So the safeguard aspect of the code goes beyond this degeneracy. Codon assignments are such that similar amino acids tend to be specified by similar codons. For example, codons of the various hydrophobic amino acids depicted as brown in this figure are clustered in the first two columns of the chart. Consequently, a mutation that results in a base substitution in one of these codons is likely to substitute one hydrophobic residue for another. In addition, the greatest similarities between amino acid related codons occur in the first two nucleotides of the triplet, whereas the greatest variability occurs in the third nucleotide. For example, glycine is encoded by four codons, all of which begin with the nucleotide GC. So this was a small video about genetic code, which discusses some important properties of genetic code. What is genetic code, along with universal decoder chart for genetic code and how to read this. So thank you for listening.